So I guess it's time to get started. So this is class number 14. So I thought today I would use mostly to go over some of the more important aspects of the previous lectures, just to kind of, rather than introducing a whole lot of new material at the end, I thought we could just go back and review some of the parts that I thought were more important. And these are the parts that may or may not show up on the final test in 2 weeks. The test is going to be pretty much similar in format to the 2 quizzes that have already gone. We've already had. There will be 15 questions. Pretty much 1 question from each lecture. Probably several questions for meiosis because there were multiple questions on meiosis. Today would also be a chance if you have any questions that you've been thinking about chromosomes during this past 14 weeks but haven't asked yet. It would be a good time to ask now because our time is coming to an end. I'm just going to go ahead and start with the first class. I was kind of giving an introduction to chromosomes as a physical object and some of the interesting physical aspects of chromosomes. In addition to the fact that they contain all our genetic material, there's some facts about them that are kind of interesting that I find. So although we've made great strides in making new memory and hard drives and everything, we still lose out to chromosomes in terms of information density. We can store a lot more information in a much tinier space using DNA than any other man-made material. So evolution is already is beating us in the information game. So our chromosomes, and it's especially interesting considering that most of our DNA is junk, or a lot of our DNA does not code for proteins, and we can't say that there's any information there. But even still, chromosomes are better than any material that we've so far designed. And also, it will last a lot longer. The information in these chromosomes will last for 100,000 years, just sitting in fossils, so we can reliably extract DNA from Neanderthal bones or other well-preserved material. And it lasts for a very long time. And also, if we think about its information lifespan in living cells, because of evolution, keeps information from getting lost, especially if the information is important. We can say that some of the information in our DNA has been around for about 1 billion years, since the beginning of, practically since the beginning of life itself. So that's one thing that I find really amazing about chromosomes. And we've only known about them for the past 100 years. So the information in them is really, really quite old, but we're still really, in some ways, just beginning to understand how chromosomes work. Because we're humans, and I want to convince us that chromosomes are important to us, understanding chromosomes is actually relevant to understanding our health. There's a little bit about its application to human biology and human disease, and it starts with the techniques used to count chromosomes and the question of how many chromosomes do we have and when did we figure that out. And you might remember that it was kind of shockingly recently that we actually knew the accurate number of human chromosomes because of a mistake, a pretty honest mistake, understandable mistake that this gentleman made when he was counting human chromosomes. He counted 48, and he counted probably several times, and he kept getting the number 48. But his technique gave him pictures that looked like this, and so probably he had to guess a lot. And there was also a problem in that there was one of our long chromosomes had a gap in it that didn't get stained by the stain that he was using. So one of our chromosomes always looked like 2, so he was counting an extra chromosome. So that's this right here. Probably he might have mistakenly counted this as 2 chromosomes instead of just 1. And it wasn't until 1956, so that's within living memory. My mom was born in the 40s, and she had heard in her elementary school class about chromosomes. She might have learned that humans have 48 chromosomes. it took a much better technique, and people were very patient and figured out ways to prepare cells so that all the chromosomes would be spread out. And that enabled us not only to get an accurate count, so now we know, of course, that there's normally 46 chromosomes in both men and women. Not just man, but that's for both of us. So 46 chromosomes. And what I wanted to point out with this number is that the actual number doesn't matter. We would have been just as fine with 48 chromosomes. In fact, all of our 8 relatives have 48 chromosomes. So 48 is a perfectly fine number to have, but it was really important to understand that 99% or so of human beings have 46 chromosomes. So if you have a different number, an atypical number, that might be related to human health. So these techniques, not only did they let us count chromosomes very well, but when these staining techniques improved, we could start to see some detail along the length of each chromosome, in addition to just counting how many there were. So the other, if you look at this guy's drawings again, I don't think he had enough detail to look at patterns within each chromosome. So he was just counting the number of chromosomes, but he wasn't attempting to say, to assign an identity to these chromosomes. But once these banding patterns became easily achievable, then we could say, okay, very reproducibly, there's this light, dark, light pattern that is the same for a particular chromosome in each cell. And that let people quantify not only do you have an extra chromosome, are you missing a chromosome, but which chromosome are you missing? And that led to a lot of identification of chromosome atypicalities or abnormalities, and the most famous one of which is the trisomy for chromosome 21. Trisomy means you have 3 chromosomes, and if you have 3 copies of chromosome 21, that most often leads to the syndrome known as Down syndrome. And having an extra chromosome from some other chromosome numbers also leads to some other developmental syndromes, but actually having an extra chromosome of most of these will lead to death, because as we learned, if you have too many copies of certain genes, then that can't support normal development, and the fetus dies before full term. When people could identify chromosomes with these banding patterns, they saw not only did people have a full extra chromosome or a missing chromosome, but also a tiny part of the chromosome could also be deleted, and that was actually quite common as well. And so every cell in the person's body would have a missing piece of this chromosome, which means that it happened in the parents, or it happened very early in development. And syndromes associated with this, with missing pieces of chromosomes, can also be quite severe. Even if you're only missing one of the 2 chromosome pairs. So in this case, you're missing all the genes located in this region of the chromosome. But that's enough to give this very typical syndrome called Cri-du-Chat syndrome, where there's also delayed development and distinctive other syndromes and developmental abnormalities. Those are somewhat rare, but it's about this one is 1 in 50,000. So it used to be that to test for these sorts of things, the progression here would be a lot of people. So this syndrome, for instance, was known well before it was known that it had a chromosomal basis. This syndrome was identified a long time ago, and with many developmental abnormalities like this, many congenital genetic abnormalities, people started to look at chromosomes and figure out that they could see, just by looking at the chromosomes, that there was something associated with it. So just based on the techniques that people had, you could only detect an abnormality if the region that was deleted was quite large. So the ones we know about from this classical cytogenetic approach, they're quite large. You have to be missing about several megabases worth of DNA for it to be visible in this sort of assay. But now we have more modern tools, and we can test for a lot more chromosomal abnormalities with this technique that I mentioned early on. It's called NIPT, for non-invasive prenatal testing. We don't need to worry about what it stands for, but it's NIPT. And the non-invasive part comes from the fact, so this is a test that's used to determine, not you yourself, but if you're pregnant with a fetus, you're testing the fetus's genome to see if it's missing any chromosome pieces. And it used to be that you actually had to go in with a needle into the mother's body and sample the fetus's own cells. And that was an invasive procedure that actually could lead to some complications of pregnancy. And so in recent years, people have wanted to do this same testing. Of course, you want to test whether your baby is healthy, but to do it in a non-invasive way. And this technique takes advantage of the fact that actually there's a whole lot of exchange going on between the fetus's body and the mother's blood supply. The fetus has a lot of cell death going on during development, and DNA from those cells actually comes out into the bloodstream and is floating around the mother's bloodstream. And in fact, if you sample the blood from a pregnant woman, you will find a lot of the DNA, there's just free-floating DNA in her blood from the fetus, and it's more than her own DNA, because there's during this development, one of the natural processes in development is cell death, and that generates a lot of broken DNA. So this free-floating DNA, instead of going in with a needle and taking it out of the fetus's body, you can take it just from the mother's bloodstream and then do DNA sequencing. And then this is really sensitive, so it's really sensitive to small deletions. So instead of having to look with your eyes and seeing that part of a chromosome is missing, you can go down the chromosome and see how many times did you detect through sequencing this part of the chromosome. And because most of the genome is represented with the same proportion, the amount of DNA you detect from anywhere in the genome should be the same. If you look at chromosome 1, chromosome 2, chromosome 3, if you're just sampling random pieces, the chance of picking up any part of the genome should be identical everywhere, except if you're missing a piece of the chromosome. So if you're missing one of the two chromosomes, then that part of the genome will only be represented half as much, and so you'll see a dip in the probability, or a dip in the amount of times you sample that. So this whole genome sequencing method is much more sensitive than using our eyes. And it's almost now replaced the older method. So this is just another way using sequence information instead of microscopy to look at chromosomes. So there was a lot of this, but then we kind of switched after the human health aspects. We started talking about physical properties of DNA, and this is everybody's favorite handedness slide, where one of the things I wanted you to be able to do is to look at a helix like this and judge whether it was right-handed or left-handed. And I hope everybody remembers that DNA is always left-handed, right? No, that's right-handed. It should be right-handed. That's all right. I think you got it. The DNA should be right-handed, and you should be able to look at other helices and do the hand trick with you point your thumb along the direction of travel, and then if your fingers curl around, matching the helix curvature, then it's right-handed. If you have to use your left hand to do that, then it's left. Okay. I might decide to test that again. It's a good skill to know, I think. You should be able to check it out. And you should be able to look at other people's pictures of DNA in press releases or articles and see if they got it right or not. So we also talked about some of the building blocks of DNA. I went into quite a bit of detail about what you call the base versus the nucleotide and the nucleoside. But I think as long as we know that the bases are A, T, G, and C, and which one pairs with which, I think that will set us up. But actually, one more piece of information is that the bases come in two types. There's pyrimidines, which are these small molecules. They only have one ring like this. And then there's purines, which are a bit larger. They have a two-ring system. It's a little bit hard to see here. Right. And yeah, so the other aspect of DNA structure that's important is that since the two bases that pair with each other, they're not sitting directly over the axis of the double helix, but they're offset a little bit. That naturally leads to two different sized grooves in the side. So there's in the side of the DNA molecule. So there's two gaps as these bases turn and go along the DNA path. The major groove, which is bigger, and the minor groove, which is smaller. And you can see more information in the major groove. So most proteins that bind to DNA will bind along the major groove because they can, by binding here, they can see which bases they're binding to. Whereas the minor groove has less information and things like DNA dyes are more likely to bind inside the minor groove. And because we have about 2 meters of DNA in each of our cells if we stretched it out. But of course, DNA needs force to be stretched out. So if a more fair comparison is if this DNA were all kind of free floating in solution, it would still be quite large. It would still be about, I don't know how big it was, about the size of a marble or something. So we would have a lot of DNA, like a handful of DNA for each cell, and that has to be squeezed into a 10 micron or less sphere inside our nucleus. So our DNA undergoes several rounds of hierarchical compaction. And the most basic and probably most important level is this first level where DNA is wrapped around this structure called the histone octamer. And together, the DNA and the histones together that are made of these 4 proteins, they make up a structure called the nucleosome. Does anybody remember about how much DNA is wrapped around the nucleosome? How many bases? Yeah, that's pretty precise. Yeah. That's probably I guess between 150, 146. I would accept anywhere between 140 and 180, depending on where you count. And there's also a little bit of DNA in between each nucleosome that I guess doesn't count for what's wrapped around. And interestingly, although DNA itself is right-handed, when it's wrapped around the nucleosome, it's wrapped in a left-handed spiral helix. And the nucleosomes look like this by an electron microscopy. Depending on how much salt you put into your solution, you might get an expanded view of nucleosomes like this, or this compacted 30 nanometer fiber view of nucleosomes on the top. And because people had seen this kind of 30 nanometer fiber view in some cells, it was thought for a long time that this represented a packed version of DNA. But now it's more and more accepted that this 30 nanometer fiber is, if it exists at all, it's only in a few specialized cell types. And mostly our DNA never takes on this form. It's really more like this, but just much more chaotically arranged. It's not spread out like this inside our cells. But this is really an important image because it was a direct physical confirmation of the theory of DNA wrapping around nucleosomes that was just inferred by the patterns that cut DNA molecules made on gels. And so DNA wrapped around nucleosomes is one level of chromosome compaction. And it also serves as the basis for packing it even further, because depending on how close these nucleosomes associate with each other, DNA can be made more compact or loose. I've used the words euchromatin and heterochromatin many times, and they're pretty important in cell biology, so we should remember them. And one of the main differences between the two is how the DNA and the histones have been chemically modified by enzymes inside the cell. So heterochromatin is most often associated with methylation. This CH3 chemical group has been added to the DNA and to histones. Methylation of DNA usually happens in mammals anyway at this cytosine, so only seeds can get methylated. And methylation of cytosines in mammalian DNA is most often associated with heterochromatin. So it's thought that some proteins come along and bind, they stick to this methylated cytosine, and those are the same proteins that bind further proteins that cause methylation of the histones themselves. And with all this methylation going on, the histones start to pack together until they're so packed that it's difficult for them, for the genes inside that chromatin to be turned on. In contrast, euchromatin, the DNA is usually unmethylated, so we don't see a lot of this methylation on euchromatin, where you have active, open, so active gene expression and open chromatin. And in fact, the histones within euchromatin are more often acetylated, which there's this acetyl group instead of a methyl group. And a picture of that I kept showing, in which you should be able to perhaps identify where is the heterochromatin and where is the euchromatin, is this electron microscopy picture, where the density of DNA is directly being measured here. So we know that there's actually more DNA sitting here than there is sitting here. So it's really the density of DNA that is important here. And does anybody, you guys remember what these circles were now? I mentioned them a few times. Yeah, that's right. And remember the main molecule that is sitting here? What is it made of? Yeah, that's right. Yeah. Yes, ribosomal RNA is made here. So with electron microscopy, it doesn't really look too different, although the density is a little different. So yeah, there's no DNA in there. So if you have a specific DNA dye, you won't see staining. Okay, yeah. So that was that's kind of the compaction and heterochromatin part. Then I was talking a little bit about chromosomes and cell division, and one of the most important regions of the chromosome, most chromosomes, is this region here called the centromere. There's two words that we use when we talk about this region. So the centromere is the DNA region. So it's the region of the chromosome that is used to recruit this other thing called the kinetochore. And the kinetochore is made of many different kinds of proteins. So we can say that the centromere is some DNA, and then on the DNA we build this structure called the kinetochore. So this is some proteins, that's the kinetochore, sitting on top of the centromere, which is DNA. And so it's kind of easy to confuse them, but they're different things. And what the job of this structure is, what the job of the kinetochore, it's where the microtubules that pull chromosomes apart can bind onto. And somehow, the centromere is usually a little bit smaller than the rest of the chromosome. And it's because the DNA around the centromere is actually packed a little bit differently. In fact, it's been found recently that centromeric DNA has a different histone. Instead of normal histone H2A, there's a different histone sitting inside centromeric DNA, which makes the DNA wrap around it a little bit differently. So the chromosome is a little bit tight around the centromere. And we talked about this signaling system that chromosomes have So when a cell divides, we know cells have to divide in order to survive and multiply. But how does the cell know when to divide? It actually knows when to divide when the chromosomes say it's time to divide. So you have to remember that part. The chromosomes know when it's time to divide when they're being pulled from both sides by microtubules. So if a chromosome is not yet being pulled, maybe the chromosomes get pulled at their centromeres in opposite directions, and that puts the DNA under some tension, which turns off this signal that the chromosomes are sending. So even a single chromosome that's not yet being pulled, like this chromosome over here, it hasn't yet entered the center of the cell where chromosomes are being pulled back and forth. So since this isn't being pulled yet, it's delaying the cell division. And it sends this signal through protein that is coming on and off the chromosome and checking whether there's tension yet. So this signal that tells the cell to wait is called a checkpoint. There are many checkpoints in the cell cycle, but we talked about the one that delays the cell division until all the chromosomes have attached. So chromosomes are very important because they direct the signal that tells the cell when to divide. And that's important. At the end, I'll talk about how that's related to cancer, because cancer cells, of course, do not listen to this signal, and they divide whenever they feel like it. And the way we know about this tension-based signal is by looking at these really interesting edge case chromosomes of the praying mantis, where you often naturally have an unpaired chromosome. Over here, there were 2 X chromosomes and 1 Y chromosome in the praying mantis. And very often, one of the X chromosomes is not attached. So here's all the chromosomes in the middle, but there's 1 X chromosome that's not there yet, and is delaying this cell cycle. And these experimenters could put a glass needle into the cell and pull on the chromosome, just put it under tension, and that led to the whole cell thinking that now it's time to divide. Even though this chromosome hasn't joined up yet, it's still over here. But using this glass needle to pull on the chromosome, you could override that signal. So this is how it was proved that chromosomes were the messengers that told the cell when it's time to divide or not. We next talked about the chromosomes in their position inside the nucleus. I'll go to the next one because this is just a close-up of the nuclear lamina. The nuclear lamina was this this meshwork, this kind of inner nuclear skeleton that gave structural integrity to the nucleus. It's also made of proteins, so this is what those proteins look like. So they make this really cool mesh kind of basket that coats the inner surface of the nucleus, and it not only makes the nucleus rigid and nicely spherical, but it also allows some chromosomes to attach to the nuclear lamina and go to the right place. And for some reason that we don't know yet, there's a mutation. There's a common, it's not common, it's actually quite rare, but when you have nuclear lamina that cannot form correctly, there's a mutation in this protein that makes it unable to make this nice circle. That actually leads to this premature aging disease called progeria. And there are other diseases that also result from different kinds of laminin mutations. So it's an important molecule for human health. Although the connection, why it is that this changed lamina results in aging, is not really clear yet. So continuing with chromosomes in the nucleus, we learned that chromosomes are not just always randomly positioned in the nucleus, so the DNA isn't just going wherever it wants to, but many chromosomes have a stereotypical position that's in part determined by how they interact with this nuclear lamina. So for instance, these are chicken cells that has, this one is human cells with D, this one is chicken cells. And in both cases, the smaller chromosomes, these really small chromosomes, in chicken they're very small, they seem to inhabit the center of the nucleus, and they don't have they don't have any propensity to bind to the nuclear lamina. The other thing we learned from this sort of staining is that each chromosome, even in an interphase cell when chromosomes are not condensed, each chromosome takes up its own little space and they don't really invade much into the other chromosomes. And these are called chromosome territories. And so, so chromosome location within the nucleus turns out to be pretty important for gene expression. And so, like we saw with that electron microscope image, so mostly heterochromatin tends to be distributed around. So red here is heterochromatin. In most of our cells, human cells, heterochromatin is around outside here of the nuclear envelope. But some people found in an interesting study that in mouse retina cells in the eye, the heterochromatin was reversed, and it was actually in the center of the nucleus. And that was a mystery for a while. And when people looked at different animals, they saw different locations. So some animals had a heterochromatin in the center, and some, like us, had heterochromatin out at the edges. And it didn't really seem to go with the phylogeny. It didn't matter which animal, like it wasn't related groups of animals that had the same organization. But as you might remember, what it did correlate with was whether the animal was nocturnal or not. So animals that need to be active at night, for whatever reason, well, they think they know the reason. Animals that are active at night and need to see in very, very dim light had eyes, nuclei that look like this. So the heterochromatin goes to the middle of the nucleus, whereas animals like us that are active in the daytime, the heterochromatin is free to go where it normally goes, which is to the outside of the nucleus. And it's thought that it actually acts as a little bit of lens to actually capture more light, prevent light from scattering around, and helps them see. So organization of chromosomes in the nucleus can actually come up in pretty unexpected places in biology. That was it for the organization. And then we spent a lot of time talking about meiosis and genetics and the chromosomal basis of genetics. So this is kind of pretty much a review of some high school biology here probably, but it's good to keep in mind some nuances that we might not have picked up back then. So for instance, we all have heard about dominance and recessiveness, things that are dominant or recessive. And in this case, there's two genes here, so these symbols refer to different genes. There's a yellow gene and a round gene. And the different forms of these genes, remember, they're called alleles. So the yellow gene comes in two different kinds. There's the big Y and the little y. And the round gene also comes in two forms, the big R and the little r. And these alleles seem to have different effects. So is the big Y, so you notice that whenever you have a big Y, these C's are yellow. So this big Y seems to always be associated with yellow C's, and the big R is associated with round C's. So it's tempting to say that the big Y then is the dominant allele of the yellow gene, and the big R is the dominant allele of the round gene. But that's actually not the way real genetics is. We talk about it because genes have many different activities in many different tissues and in different contexts. So a gene or an allele itself cannot be dominant or recessive, or it doesn't make sense to talk about it being dominant or recessive without specifying the actual context. So really we don't say that the genes or the alleles are dominant, but we can say that the yellow phenotype is associated with this allele. This yellow C phenotype is associated with a certain allele of that gene. So then we talked about Mendel's laws a little bit, which are not really laws as such. They're not physical laws by any means. They're just some things that he saw in pea plants, but they turned out to be very important. So by looking at the way things segregated, he was able to deduce that, of course, each gene comes in 2 copies. So the yellow gene comes in 2 copies in each cell, and the round gene comes in 2 copies in each cell. So each diploid cell has 2 copies of each gene. But then when they make gametes, when they make pollen in his case, or sperm and eggs in our case, these 2 copies of each gene go only in 1 copy to these haploid cells. So his first law was that these 2 alleles come apart from each other. These 2 genetic elements must somehow come apart and end up in different haploid cells. His second law that he saw, because of the way these round and yellow and other genes behaved when he looked at them, he saw that they never influenced each other. So the roundness of a seed never had any influence on the color of the seed, at least with the genes he was looking at. So that led him to propose that these genes were all independent. So in his vision, the genetic material was this bag of genetic elements that somehow they knew that they had to go away from their partner, but there was no linkage between these genetic elements, that each one was free to go wherever it wanted to go. Now we know that the second law is not really a law, it's just a statement of what can happen if genes are on different chromosomes. But if genes are on the same chromosome, of course, then they don't necessarily show this second law behavior. So it's really not good to call the second law a law. It's more of a it's more of a suggestion. So let's skip that one. So this is the evidence that kind of breaks his second law of independent assortment. So instead of seeing if these yellow and round and this other pod color gene, if they were truly independent, then if you crossed this combination of genes with this combination of genes, then you would get all sorts of combinations in equal numbers, because each of these Y or Y, big Y, little y, big R, big P, could go independently of each other, and all these numbers would be the same. But in fact, what he found was not that what he found was that there's, what later geneticists found was that there's strong evidence that these genes are linked together in a certain way, but they're not perfectly linked together either. So if they were perfectly linked together and never were completely dependent, then all you would see is this combination with all big letters, or this combination of all small letters. But in fact, so they're not completely independent, but they're also not completely linked. And that was a kind of confusing observation at first, because people, I think they were really hoping that they would find, yes, genes are located on chromosomes, and they just always go together. But it was known already that sometimes these genes would recombine with each other, and that's what this data ended up showing. So by looking at the number of cases where you got, for instance, the big Y, big R, and small p, then there had to be some exchange at this point between the 2 chromosomes. And by looking at the relative rates at which these exchanges happen, people could start to place genes on actual positions on chromosomes. Anyway, this kind of data became the basis for this chromosome theory of inheritance. It's kind of strange to us. We kind of take chromosomes for granted, and that there was ever a chromosome theory seems a little bit weird, but of course all this stuff had to be reasoned out and proved. And so it was by looking at how often these genes recombined with each other that provided this kind of evidence that genes were in order along chromosomes. It's actually kind of fortunate that genes did show this recombination behavior, or else we would never probably have figured out that there is a definite order and with different distances between genes. right? So yeah, these features of this chromosome theory. The most basic one, of course, is that chromosomes are genetic material, and that was found just by a lot of associating between what chromosomes look like and what features you inherited on those chromosomes. And this coming apart of two alleles that Mendel saw was exactly the same as the coming apart of the maternal and paternal chromosome pairs. And independent assortment, the second law is a law if the genes are on different chromosomes, so that part of his law was correct. But if they're on the same chromosome, then they're not independent. So this new information didn't really alter what Mendel saw, it just kind of expanded on it. And then the absolute best evidence for this was seeing this actual picture of what had only been theorized before based on genetics, that chromosomes actually do this recombination. Just by looking at different colors of seeds or leaves or pea pods, people knew that chromosomes must be exchanging information, but then it was through this sort of picture that people could prove that it was actually happening. All right, so then the next bit is talking about the stage in biology when this stuff happens. So the genetics is kind of the study of the recombination of these heredity factors. But looking at it from a cell biology perspective, we could get into the study of meiosis and what chromosomes are doing in order to form the basis of genetics. So meiosis is really the cell biology basis of genetics. And in order for chromosomes to be able to exchange information, they have to do a lot of things first. They first have to find each other, they need to pair, and they need to synapse. So synapsis and pairing are two different things. Remember, pairing is just chromosomes come together into the same space, whereas synapsis means that they get this synaptic complex protein. Let's see. This polymerization of this ladder-like protein that connects the maternal and the paternal chromosomes together. So those things need to happen. Remember I showed that there was this kind of mathematical difficulty with pairing because as you add more and more chromosome pairs, it becomes more and more difficult for any kind of random process to bring these chromosomes together. So I talked about the fact that pairing has to be kind of enforced or actively promoted by the cell, and there are some ways to move chromosomes around to bring them together. Okay. And then so once chromosomes are paired, then how do they do this exchange of genetic information? We're kind of getting into material that we've covered a lot, so it should be maybe looking more familiar. But remember there was this enzyme called SPO11 that somehow decides it's going to cut the DNA. And even this first step of cutting the DNA doesn't happen randomly, but it's directed to certain places by the landscape of chromatin. So it's more likely to cut in an open euchromatin region, for instance. So after SPO11 cuts, it makes this DNA double-strand break, which means that it cuts both strands of DNA. Then there's a bunch of other things that happen I won't test too much about, but there's this one structure that forms called a D loop, where one strand from the broken chromosome invades the other strand. And if one DNA strand comes in and invades, this DNA is only a double helix, not a triple helix. That means that this other DNA strand that was there first has to come out. So when it comes out, it's sticking out to where the other chromosome was, because this kind of looks like the letter D on its side. This is called a D loop. So this D loop is involved in connecting the broken chromosome with the unbroken chromosome, and that happens naturally as this broken strand comes in and DNA replication causes it to get longer and longer. It makes the D loop also get longer and longer until finally it's long enough to encounter the other broken strand, which has been sitting here. So these two broken strands make contact with the unbroken strand through this formation of a D loop. And that finally gets repaired into this structure. And this, of course, is the double Holiday junction that we've talked about a lot. That needs to be, because this is a bubble in this DNA that is still connecting these two DNA molecules. When these chromosomes divide, when they come apart, this double Holiday junction also it needs to be resolved. It needs to be taken apart. There's two ways this can happen. You can cut these junctions in opposite senses, which leads to non-crossover based on the way that these DNA molecules are arranged. Or you can cut them, this is a little too much, you can cut them in the same sense, and if you cut them in the same parallel orientation, that's what leads to crossovers. So it doesn't matter which parallel orientation you cut them in, they will both lead to crossovers. But in all cases, whether it's a crossover or a non-crossover, there is always some small region that contains DNA from both strands. So even if you don't cross over, even if the original chromosome maintains its integrity throughout the entire this Holiday junction region, there's always a little bit of DNA that gets, so there's a little bit, a little hard to see, but there's a little bit of green DNA mixed in with this blue DNA strand. So even if there's not a crossover, there's still some genetic information that can come along for the ride. Okay, and that's what it looks like as a close-up. Okay, so then we talked about some consequences of this asymmetric female meiosis. So in male meiosis, all four sperm are are equivalent. So there's no competition among these chromosomes to get into for which sperm they get into. From the chromosomal point of view over evolution, neither of these chromosomes care if they get into this sperm or this sperm or this sperm. So in male meiosis, it's relatively straightforward, and the chromosomes just divide, and that's the end of it. But in female meiosis, because the whole definition of being female is that they make large gametes, and in addition to this large gamete, there are smaller products called polar bodies that just get thrown away. In this case, from the chromosome point of view, there is some competition. So if a chromosome can do anything that increases the odds of it getting into this product, then it will do it. Okay, even if that thing raises the probability by a fraction of a percent, even if it's only a very tiny effect, over generations and generations, that will get magnified. And so there's this competition between the chromosomes for which one is going to get into this female gamete, and that leads to this idea of genomic conflict and meiotic drive. So competition to get into the oocytes, into the egg, can lead to situations like this, where here we've got this unbalanced situation where somehow the red chromosome, or maybe even just one allele on the red chromosome, has figured out a way to beat the yellow chromosome and get into the oocyte more often. So here's only 4 cases where the yellow chromosome has gotten in, and another 10 cases or so where the red chromosome has gotten in. And the effects we see are about that size. And one of the most common manifestations of this that can be seen in rare cases where chromosomes break is called the unequal centromere number rule. And that is that if there's any imbalance in this cell division, this first female meiosis cell division, such that there's n chromosomes on one side and n+1 chromosomes on the other side, then because of this conflict, there's this strong bias for either the greater or the fewer number of centromeres to go into the egg. And it can go either way, as we've seen by comparing, for instance, humans and mice and chickens. In mice, it goes one way. So in mice, it so happens that the side with the greater number of centromeres, so where you have the broken chromosome, there's a strong bias for the broken chromosome with 2 centromeres to go into the egg. So this is the number of times that this chromosome was recovered in the egg, and this is the number of times that the unbroken chromosome was recovered. So this is a pretty strong bias that you're much more likely to get the broken chromosome in the egg than the single chromosome. And likewise, where actually the opposite is true in humans and chickens. So in humans, if one of our chromosomes break, then the unbroken chromosome has a strong bias to successfully get into the egg. So it seems like humans and mice have this opposite tendency to where I guess you can say that human chromosomes have this tendency for smaller numbers of centromeres to be inherited, whereas mice have a tendency for larger numbers of centromeres to get inherited. And that has led to a pretty interesting phenomenon that maybe I don't have a slide about, but I'll try to recall it for you, that human chromosomes therefore mostly look like this, with the centromere in the middle, whereas mouse chromosomes usually look like this, with the centromere at one end. This is because even though the breakage of a chromosome is a fairly rare event, this bias is so strong that whenever a chromosome does break like this in mice, it will be much more likely to get into the egg. So over time, even though chromosome breakage is rare, you will accumulate a species that consists entirely of chromosomes that are broken at their centromeres. And the opposite way can also happen. So this mouse with centromeres at only one end, sometimes these chromosomes can fuse together just because. There can be a break in the DNA and they will just fuse together. So even though that's a rare event, if this symmetry could get flipped, then eventually over a certain number of generations, you would end up with a karyotype where you only have centromeres in the middle. So I showed you this slide where people predicted the theoretical distribution of centromeres in the middle versus the end, but the actual distribution that we see is completely opposite of that. Okay, so there's this meiotic drive that happens because this asymmetric meiotic cell division. Okay. So the next topic was evolution of chromosomes. It has a lot to do with this topic, in fact, with the previous topic. So chromosomes, chromosome numbers, if you scan, you make an atlas, as people like to do, early in the last century, of how many chromosomes different organisms have. The numbers can be all over the map, from one pair of chromosomes to hundreds of pairs of chromosomes. And since we all share this evolutionary tree, that means, of course, chromosome numbers can change over time. This is one of the main ways in which that number can change over time, is these chromosomes breaking and fusing. And we have a very nice case of this in humanoid or hominid chromosome evolution. So if you look at the family tree of our most recent 4 or 5, so these are called the great apes, I guess, including us. Look at the most recent tree of the great apes. You can see the chromosome numbers change. So this is the haploid chromosome number. We have one fewer than all the other apes. That doesn't make us special, of course. It just means that that something happened somewhere along the lineage to make it humans. So it's not that we lost a chromosome. We have all the same genetic material that all the apes have. So every ape in a chimpanzee has every gene in a chimpanzee has a corresponding gene in a human. We haven't lost any material. It's just, and they haven't gained any, but actually, yes, chromosomes fuse together into a single chromosome, and that happens to be human chromosome number 2. And depending on the source of which ape that came from, I think in our most closest, I think this is chimpanzee, gorilla, and orangutan. So bonobos are not listed here. But chimpanzee, just 2 chromosomes fused together. In the gorilla, these are also 2 chromosomes. I guess in the orangutan, it's 2 chromosomes plus a little bit of another chromosome that got translocated at some point. All of our other chromosomes, interestingly, line up pretty much 1-to-1. So even though we have this drastic change from the other apes, all of our other chromosomes, down to the smallest ones, are very highly conserved. And this is just a schematic of and this is chromosomes, and this is what they actually look like. So this is a pretty cool study that was done a while ago. So and this the mechanism of this fusion. It's slightly different though. So the mechanism of this kind of fusion is different from this. So these were not chromosomes that had a centromere at one end that fused together into chromosomes with a centromere in the middle. You see, each of these chromosomes has its own centromere, and we kept genetic material on both sides of both centromeres, which means these chromosomes didn't break at their centromeres. They actually fused together somewhere here near the end. So those chromosomes fused together, and then one of the 2 centromeres, I guess this one, became inactive. So the centromere lost its activity. I know it's this one that lost its activity because here you can see this pinched region here indicates the centromere. So this centromere stayed active, but this centromere was lost. So it's slightly different than this mouse process. But it did lead to a more central centromere. So our chromosome 2 centromere is more central than either of those 2 centromeres. So there might have been some meiotic drive involved, but I'm not convinced there was. Okay, so after that, the next topic was sex determination, and we learned there are many different ways that different animals determine sex. So most mammals use what's called this canonical XY system, where it's the presence or absence of a Y chromosome that's responsible for the sex determination. So no matter how many X chromosomes you have, it doesn't matter. What matters is whether you have a Y or not. So XX individuals are female sex, XY individuals are male sex, but then adding more X chromosomes doesn't make any more femaleness. It's just having a Y chromosome or not that in most cases leads to male development. And I'm pretty sure that's true for almost all mammals, but there could be some exceptions. And the reason I mentioned extra X chromosomes in addition to a Y chromosome is because this actually does happen from time to time. So although I mentioned that this chromosome 21 triploidy was very common and it gave rise to some developmental abnormalities, the X chromosome and even the Y chromosome too are much more tolerated if they have higher copy numbers. So XY individuals, there are some health problems, but they're not actually that severe in either of these cases. So female with only a single X chromosome can also be well tolerated, and this leads to what is called Turner syndrome. There are some health problems associated with it, but it's not really that severe. And having extra Y chromosomes actually doesn't do anything at all. Males who have XYY karyotype usually don't even know that they have it. It only comes up if you do a chromosome test. That's the same is true if a female has an extra X chromosome, they usually don't know that they've got an extra X chromosome. So although chromosome 21 is a very tiny chromosome, and that's one of the reasons why having an extra one can be tolerated, the X is actually a pretty large chromosome. It's got thousands of genes. It's a big chunk of the genome. but it can be tolerated much better than any other similarly sized chromosome. And the reason, we talked about the reason for that, but I'll get to that in a bit. So in addition to the mammalian, so this XY system is very familiar to us, but it's by no means the most common or most or simplest. It's actually quite a complicated system. But birds and some other insects like butterflies use a different system called the ZW system. And so say that there's nothing intrinsically different between the XY and the ZW system in terms of the chromosomes themselves. There's no such thing as a Z chromosome that you can identify by looking at it and say, oh, this is a Z chromosome, this is a W chromosome. Those names don't really mean anything. This is called ZW just to distinguish it from the XY, because in the ZW system, males have two identical chromosomes. So we say males are homogametic, meaning that all sperm produced by males are the same. All gametes produced by males are the same, whereas females are the heterogametic sex in the ZW system. So females can lay two different kinds of eggs. There's eggs that have a Z and there's eggs that have a W. So in that's the opposite of the XY system. So in which males make different kinds of sperm, but females make only one kind of egg. So it's more precise to say that the XY system is really the heterogametic male system, and the ZW system is the heterogametic female system. Okay, so there's nothing special about the Z versus an X chromosome. And here, it's in this system, like the XY system, it's whether or not you have this heterogametic, this different chromosome, this W chromosome. Having a W chromosome makes the individuals female in all cases. So the next thing was the kind of crazy system that the platypus has, to show you that there are more complicated systems in mammals. So it is kind of an XY system, but it's the X and the Y are actually made of multiple chromosomes each. So what you call the X chromosome in a platypus is actually a group of 5 chromosomes. And similarly, what you call the Y chromosome in a platypus is a group of 5 Y chromosomes. And these chromosomes are usually separate from each other during most of development, but during meiosis they actually bunch together and travel as one group. They don't recombine with each other, but they line up as if they were a single chromosome, and that's how they stay together in this group. Right, so that's how the platypus manages to keep its X's and Y's going to opposite poles during meiosis. How humans do it, though, we only have a single X and a single Y that are very different from each other. So how do they manage to get inherited normally? And that is based on the question from the last quiz, by using these PAR regions, or these pseudo-autosomal regions, which, although for most of the chromosome's length, the X and the Y are very different from each other. They have almost nothing in common. There's only a few genes on the Y chromosome, whereas there's many, at least 1,000 genes on the X chromosome. However, these PAR regions are actually the same. They're just completely identical. And they maintain their identity by recombination over meiosis. And that's actually their function, is to recombine during meiosis. So although the X and Y chromosomes are very different, they're not like chromosome 1, which is pretty much the same whether you get it from the mother or the father. The X and the Y chromosome manage to find each other and recombine at this pseudo-autosomal region. And that's how they can make a connection which lets them go to opposite daughter cells. Spent a little time on the C. elegans system, where it's just an example of it's also the male is also heterogametic in the C. elegans system, but here they don't have 2 chromosomes. So it's kind of like the XY system, but instead of the sex development being determined by the presence of a Y chromosome, it's actually a lot more complicated. It's determined by the ratio of the number of X chromosomes to the number of complete sets of autosomes. So in this case, you've got two sets of autosomes. Here's one set, here's the other set. And you have two X chromosomes. Therefore, the ratio is 1, and that leads to hermaphrodite. Whereas if you have only a single X chromosome, then the ratio is 0.5, and that leads to a male. We discussed, although the story was a little bit complicated, but the reason why it's the ratio and not just the number of X chromosomes was determined by, they made worms that had different numbers of X chromosomes, so 3 X chromosomes or just 2 sets of autosomes. And they saw that depending on that ratio, depending on the actual ratio you have, there was some cutoff where the development flipped from male to hermaphrodite development. For the purposes of this, I think the main thing to know is just that in this class, all you need to know is that an individual with 2 X's is a hermaphrodite, and an individual with a single X turns out to be a male. So this could be called the XX versus XO system. As opposed to the XXXY system. XO the O just means there's no chromosome there. Okay, and then as a consequence of having this heterokaryotypy, of having different kinds of chromosomes, there's also this problem that you have a different number of genes in each cell, depending on how many chromosomes you have. So an XY individual only has, let's say if the X chromosome has 1,000 genes, then they've got 1,000 of those genes. They have 1 copy of each of those 1,000 genes in each cell, whereas a female individual would have 2 copies of each of those 1,000 genes. And as we saw, with even having a single extra copy of chromosome 21, so you have 3 copies instead of 2 copies of those genes, that can lead to some pretty profound developmental changes. So how do you develop male bodies and female bodies that do not have such profound changes? You do that through this mechanism called dosage compensation, which equalizes the amount of RNA, or equalizes the amount of transcribed genes you get from the X chromosomes in either sex, or both sexes. And there's many different ways of doing that. The mammalian way is, again, it's probably the most complicated. So I would have to say mammals have a very simple sex determination system, but a complicated dosage compensation system. Because what mammals do is they do dosage compensation in females in the homogametic sex by picking one of these X chromosomes at random and turning it off. That leads to a situation called mosaicism. So the female bodies are mosaics, where some cells have one X chromosome on, and other cells have the other X chromosome on. And that leads to things like if the X chromosomes have coat colors or fur colors or other genes on them, that will lead to a mosaic of development. This is something interesting that I learned during preparation for this class. I always thought of it as simply one of the 2 X chromosomes was turned off, but it's more accurate to say that all but one of the X chromosomes are turned off, because if you have 3 X chromosomes, if you're a triple X female individual, then those cells have 2 inactive X chromosomes, and that's why XXX females don't even know that they are XXX, because in terms of, as far as gene expression goes, they're about the same as a normal XX female. And the condensed inactive X chromosome ends up attached to the nuclear envelope like most of the other heterochromatin, and it makes this structure called the R body, which is the inactive X chromosome. So yeah, that kind of that's I guess that's where I'll end. I'm not going to talk about the microscopy stuff. I'm not going to be having you wear red cyan goggles and looking at the test to deduce anything. So the microscopy stuff is not going to be on the test. I'll just end here with a little bit more information about links between chromosomes and cancer, because I think it's also important to understand how chromosome breakage or chromosome abnormalities are associated with cancer. It's just one tiny little coda to the end of this class. The reason that chromosomes and cancer are connected is because sometimes, it's a rare event, but sometimes we've seen many cases of chromosomes breaking and refusing in different ways. That's called the chromosome translocation. And in some cases, of course, since a chromosome breaks, sometimes it breaks right in the middle of a gene. Then when you reform the gene, you can actually change the way that gene works. So you can, chromosome translocations can lead to a mutation where this gene actually has some phenotype in the cells. So of the super class of chromosome translocations, there's some cases where you mutate a gene, and then of those cases, there's some of those cases where the effect of that is to lead to increased cell growth. The most dangerous kind of increased cell growth, of course, is if you are disrupting one of these checkpoints. So that's one of the reasons this checkpoint for cell division is so important. The cell waits for all the chromosomes to be ready before it divides. But of course, this checkpoint is a complicated process involving many genes. And if you mutate one of those genes and you start ignoring that checkpoint, then the cell will divide before it's ready. And what can happen then is a chromosome can get caught in this cell division, and then you can lead to another broken chromosome, and maybe that will cause another gene mutation, and that process can actually get carried away, and the genome will start to change in unpredictable ways. And so you can accumulate enough mutations that eventually not only do they keep dividing, but the body loses its ability to stop that, and that's one of the ways cancer can get started. One of the most clear connections between chromosome translocation and disease is a particular kind of leukemia, chronic myelogenous leukemia, caused by what has been termed the Philadelphia chromosome. So this Philadelphia chromosome is a translocation between chromosomes 9 and 22. So chromosome 9 is a medium-sized chromosome. 22 is a very tiny chromosome. And when this translocation occurs, chromosome 22 gets even shorter. So it loses a lot of its material that gets stuck on chromosome 9. And a tiny bit of chromosome 9 material comes onto chromosome 22. And this was found by somebody looking at a lot of cells from cancer patients, a certain kind of cancer. It was a guy's PhD project to study the chromosomes of these people. And just by looking carefully, he saw this kind of abnormal chromosome, this really small chromosome that didn't look like any of the other chromosomes that he had seen before. And this is from the 1950s, just after some techniques for looking at chromosomes had improved and people could start doing this. People weren't only looking at chromosome abnormalities for these congenital birth problems, but also for cancers for a very long time. And so what happened in this case, this Philadelphia chromosome, I think, is drawn much larger than actual. It's actually really quite tiny. So a little bit of chromosome 9, so a little bit of chromosome 22 has gone on to chromosome 9, and chromosome 22 gained some of chromosome 9. And on this chromosome 22, there's this gene called ABL, or ABL, and ABL is a kinase. It's a protein that puts phosphate groups on other proteins. So I've talked about that before in context of this gene in C. elegans that removes those phosphate groups. But this protein does the opposite. It puts on these phosphate groups, In doing so, it activates proteins involved in cell division. So normally this ABL kinase is doing its job in the right place at the right time and promoting cell division on a normal schedule. But if you break the chromosome right in the middle of this gene, so yeah, if you break this chromosome and put it next to this other gene, this BCL gene, you just ran by chance, that makes this ABL kinase into a hyperactive protein that's always on and always wants to promote cell division. So it doesn't listen to the cell cycle anymore. It's just always saying, yes, let's divide. So just by this random chance, this kinase becomes hyperactive and leads to cancer of this particular blood cell type. And so when you learn about these chromosome translocations, so many of them are known by now. In fact, there's a lot of these translocations now have been mapped. So translocation between chromosome 8 and 14 is associated with Burkitt's lymphoma. You can look at all these different, these are not all cancers, of course. There's Down syndrome, chromosome 21. But you see lots of leukemias and lots of OMAs. And L is a leukemia, and this is a leukemia. So there's lots of cancers associated with chromosome translocations. And so you might think, well, are chromosomes predisposed to break and rejoin at the sites of cancer-causing genes? Are chromosomes more likely to translocate at places that cause cancer? I think the answer is no, not at all. It's just that these are the cases where people are studying chromosomes. So clinicians and researchers are not studying the chromosomes of healthy tissue from healthy people. We probably have chromosome translocations in all of our, in some parts of our bodies, but they're not leading to cancer, hopefully. So it's only these cases that have been well studied that, so there's this observational selection effect that leads to us knowing that a lot of these chromosome translocations can cause cancer. But I think it's always better not to have translocated chromosomes, so that's why it's important to stay out of the sun and don't expose yourself to gamma rays and don't eat poison, things like that. Okay, so that'll do for the review and this little connection between chromosomes and cancer I want to get across. So that's it for what I'm presenting. In 2 weeks, remember we don't have class next week. In 2 weeks we'll meet again for the last time, and we'll have a test which is not much longer than the quizzes we've been having so far. And everything on the test will be something. I won't introduce anything new on the test that wasn't discussed today. And I'll put this PDF version on that website as soon as I can, hopefully right when I get back to my lab. But if you have any questions, then please feel free to contact me.